Hello, I'm Dr. DeBellis, and I'd like to talk to you about the neurophysiology of synaptic transmission. So, now that we've explored the graded potential, which occurs in the cell body and dendrites, and we've also discussed the action potential, which occurs in the adjacent axon, we're going to move on and talk about synaptic transmission, which occurs in the axon terminals of the neuron and results in the release of a neurotransmitter into the synapse. Recall that the action of a neuron is akin to a soccer ball that bumps some dominoes, then bumps another smaller ball off of the edge of a desk. So the soccer ball, and as you see in my video for chapter 3.1, when the soccer ball pumps up, that's similar to the buildup of electrostatic pressure that happens through graded local potentials in the dendrites in the cell body. And once enough of those local potentials combine to depolarize the cell body to negative 40 millivolts, that's akin to the soccer ball bumping the dominoes, leading to what we talked about as the action potential, which is the self-propagating electrical signal that travels in one direction, in this case from left to right, and then bumps this little yellow ball here at the end. The ball rolls off of the desk, which is sort of a metaphor for the neurotransmitter being released from the axon terminal into the synapse, where it's going to make its voyage to a receptor, where it's actually going to initiate the process again. So in order to explore this last step, I want to remind you, hopefully you recall that information flows within a neuron via electrical signals, while information passes between neurons through chemical signals in the synapse, and these are neurotransmitters hitting the postsynaptic membrane. Now the synapse, and you can see the synapse is pink there, it's a junction between two nerve cells, which is essentially a small gap across which impulses pass by diffusion of a neurotransmitter. The presynaptic membrane is the cellular membrane of the axon terminal that faces the receiving cell. But the postsynaptic membrane, on the right there, is the membrane around the receiving dendrite, which has receptors that respond to chemical messages from the presynaptic membrane. So, now a neurotransmitter, which is going to be one of the main topics of this discussion, a neurotransmitter is a chemical messenger which sends messages between neurons across the synaptic cleft. So there we see that red circle. That's actually dopamine. Dopamine's a neurotransmitter. It's a biogenic amine, and we're going to talk about it in more detail, but it diffuses across the synapse, and then it activates a receptor when it reaches the other side. And whenever the receptor is activated, it actually leads to a graded potential. What happens is the receptor allows an ion to enter. And here we can see it's a positive ion. We're going to call that a cation, if you remember the difference between cations and anions. Now, one cation is not going to make all the difference in the world, but many of them in combination with each other can actually lead to something as pivotal as an action potential if the axon hillock is pushed to a threshold of negative 40 millivolts. Now, after the neurotransmitter activates the receptor, there are two things that can happen to the neurotransmitter. One thing that can happen is it can be degraded, which means that it can be broken down by enzymes. So it can be broken into its parts. It can also, so there we have the degradation of a neurotransmitter. The other option, which is a little more eco-friendly, we can recycle. And that is referred to as reuptake when we're talking about neurotransmission. In the case of reuptake, after the neurotransmitter hits the receptor, it returns, where it will take a break in the presynaptic neuron. There's actually a protein in the cellular membrane. For example, for serotonin, there's a protein that's in the presynaptic membrane that's called SERT, or serotonin reuptake transporter, and it transports the serotonin back up into the presynaptic terminal where it has a break. And the net effect is you're going to have less of the neurotransmitter in the synapse when it's reuptaken. It's almost like you're taking a basketball player out of the game and letting it have a break before it returns. So this is called reuptake. Oftentimes, the same neurotransmitter can be degraded or reuptaken at the same time. So why does the presynaptic membrane actually release a neurotransmitter? What causes this? In order to understand this, it would be helpful for us to look through a microscope at the distribution of molecules inside of inside of the intracellular space and the extracellular space. And here's a diagram you're probably familiar with. Here we have all the sodium in the extracellular space. Above is the extracellular space, and then we also have potassium. We have some protein distributed in the extracellular space, along with calcium. Notice we don't really have any calcium in the intracellular space. We have lots of protein in the intracellular space as well as potassium, but we don't really have any calcium. So what ends up happening is whenever there is an action potential, in the axon terminals, the cellular membrane is studded with our final protein channel, which is the voltage-gated calcium channels. And here is a neuron at rest, and here we can see the voltage-gated calcium channel. And we can see the extracellular, or the intracellular space right here, negative 60 millivolts. At negative 60 millivolts, the voltage-gated calcium channel is closed, and we can see the extracellular calcium here, that yellow circle. But whenever an action potential occurs, and if we look down at the lower left-sided corner, we can see that the electrical potential of this neuron is changing. It's having an action potential. Whenever we finally reach negative 40 millivolts, which is going to be the action potential, this voltage-gated calcium channel opens. It detects the voltage change during the action potential and allows calcium to enter the intracellular space. So there we see the calcium is passing through. Now it's in the intracellular space. And whenever the action potential passes, the intracellular space returns to resting potential, and the voltage-gated calcium channel closes. So what does the calcium do once the calcium is drawn in? So the calcium is drawn into the intracellular space, again, by the low intracellular concentration of calcium, as well as the negative charge of the intracellular space. Now we haven't talked yet about this, but inside the presynaptic membrane, there are pouches we call vesicles, and they enclose neurotransmitters and protect them from being destroyed by enzymes. So I liken the vesicles to when you go to a vending machine to get yourself some potato chips. It would be really disappointing if you paid for your potato chips and just spit out a bunch of potato chips. We expect the potato chips to be in a small bag. The small bag protects the potato chips and keeps them fresh. It's kind of like a vesicle is for neurotransmitter. It encloses the neurotransmitter and protects them from enzymes in the intracellular space that would actually destroy them. So inside of the presynaptic membrane are these pouches, these vesicles that enclose the neurotransmitter. And here we see the calcium has entered. And inside of the presynaptic membrane are these pouches. And so inside of the presynaptic membrane, calcium searches for vesicles. And once it finds one, it actually transports it to the presynaptic membrane. So once calcium arrives at the vesicle, it works like a tugboat. So once calcium arrives at the vesicle, it works like a tugboat, drawing the synaptic vesicle closer to the cellular membrane. And when the vesicle contacts the presynaptic membrane, they fuse, and the contents of the vesicle are released into the synapse, a process we call exocytosis. Then the neurotransmitter diffuses across the synapse, then the neurotransmitters diffuse across the synapse where they will serve as a ligand. So a ligand fits specific receptors exactly. It's like keys shaped for locks that have the potential to activate or block the receptor. So it binds to a central metal atom to form a coordinated complex. And neurotransmitters are ligands, which fit specific receptors exactly, like keys shaped for locks, that have the potential to activate or block the receptors. So there goes our ligand, our neurotransmitter, and it's going to hit a receptor. Now, one ligand's only going to have a minor local potential, but assemblies of ligands, huge numbers, can cause enough change in the charge of the dendrite to actually cause an action potential. A little bit more about ligands. Ligands are classified by the location from which they originate, either inside the body or outside of the body. So up to the right, we can actually see a dopamine molecule. Dopamine is actually produced inside the body. We're going to learn that the dopamine is produced in the midbrain and the basal ganglia. Below, to our left, we actually have Prozac. Prozac's not produced by your body. It's produced, it's manufactured. So we would say that dopamine is an endogenous ligand. It's a neurotransmitter that's produced within the body. Exogenous ligands are drugs and toxins produced outside of the body. So here we are again, taking a microscopic look into the membrane, the intracellular space, the extracellular space. Postsynaptic potentials are brief changes in the resting potential. We have two types. We have EPSPs and IPSPs. EPSPs are excitatory postsynaptic potentials. They produce small local depolarization, pushing the cell closer to threshold. So things that make us more likely to have an action potential would be excitatory. Inhibitory postsynaptic potentials, IPSPs, produce a small hyperpolarization, pushing the cell further away from threshold. So inhibitory postsynaptic potentials often result from chloride ions, and you probably remember those were the dark blue colored circles in the extracellular space. When they enter the cell, they make the intracellular space more negative, so they hyperpolarize it. So a number of postsynaptic membrane receptors, the number varies in response to lifestyle changes, such as drug use. Here we have some ligands that are headed toward receptors. Upregulations, an increase in the number of receptors, and these circles over on the postsynaptic membrane are supposed to be receptors. And so there's an increase in the number of receptors in response to decreased activity at a postsynaptic membrane. So if somebody does not have enough dopaminergic activity, maybe there's not enough dopamine, you're going to have an increase in the number of receptors. So it's a homeostatic response to the decreased activity at the postsynaptic membrane. Downregulation is a decrease in the number of receptors, and it's typically a homeostatic response to increased activity at the postsynaptic membrane. So in this particular case, if we're thinking of dopamine, for instance, if we have too much dopamine activity, too much dopamine, as a result, dopamine receptors will actually diminish in number, and then we end up with a much smaller number. So when would this happen in a human being? Well, research shows prolonged use of recreational drugs such as MDMA, also known as ecstasy, or methamphetamine can actually downregulate dopamine receptors. And this is probably the reason that many people report an inability to experience the previous level of pleasure whenever they're in the initial stages of recovery. So they have a downregulation in dopamine receptors, along with other receptors. Like with MDMA, you're probably going to also have diminished number of serotonin receptors as well. Now, postsynaptic receptors control ion channels in two ways. We have ionotropic receptors and metabotropic receptors. Ionotropic receptors open when bound by a transmitter, and they allow an ion to enter. So there's our ligand, our key right here, and this red thing here is an ion. We're going to say it's a cation. And what does a ligand do when it finds the appropriate receptor? It goes in and it opens up the receptor. And that's how these ionotropic receptors work. They open when bound by a transmitter and allow an ion to enter. And there we see the ion making its adventure, going for a vacation to the intracellular space. And whenever it gets there, notice this pink color here. I'm using that to represent the depolarization. This is a small graded potential. Again, this is just one cation. This is not assemblies of cations. And this leads to a small graded potential. So there it is floating in the intracellular space. So now that we've talked about how the ionotropic receptor works, we're going to talk about the metabotropic receptor. The metabotropic receptor is much slower. A metabotropic receptor recognizes the transmitter, but instead of opening its own channel, it activates a G protein. And this is the G protein down here. I made it a G. I don't know that they look like Gs, but it's released whenever a neurotransmitter hits it, and then the G protein travels through the intracellular space, and then it reaches a channel that it opens, and that channel allows an ion to enter into the intracellular space. And then the pink color again represents the graded potential. So, in summary, an action potential travels down the axon to the axon terminal. After that, voltage-gated calcium channels open and calcium ions enter. Synaptic vesicles fuse with membrane and release neurotransmitter into the synapse. And again, this fusion is caused by the calcium ions that drag the vesicle to the presynaptic membrane. Then transmitters bind to postsynaptic receptors, and they can cause excitatory postsynaptic potentials or inhibitory postsynaptic potentials. Sometimes these actually cancel each other out, ensuring the resting potential. Now, the excitatory postsynaptic potentials or inhibitory postsynaptic potentials, they spread toward the postsynaptic axon hillock. And transmitters inactivated or removed, so it's either degraded or reuptaken after its brief action. And then finally, the transmitter may activate presynaptic autoreceptors, decreasing its release. And what that looks like is sometimes when neurotransmitters are released, they have a negative feedback loop where they go back to receptors that actually reduce their output whenever they're being made at too high of a rate. So thank you very much. That's how synaptic transmission occurs. In the next lecture, we're going to focus more on what happens in the synapse. We're going to talk about how drugs work, how different neurotransmitters work. So thank you very much.